My title initially says Protocols to Promote Prolonged and Fast Recovery and Plasticity with TRNS. Without plasticity, but I added plasticity to it because we're actually working on learning and plasticity doing our work, both with patients and healthy subjects. So, sorry, see if I can start. Okay, so we typically live in a cluttered environment, and everyday activities require effort to be performed. They all require concentration. So from tracking our kids at the playground to driving the car, performing grocery shopping, reading the newspaper, making coffee, playing sports, studying and participating in Zoom meeting, of course, all require efforts. So let's say that ideally we would like to maintain a healthy, so I'm just checking, are you hearing me?
Yes.
Yes.
Okay, sorry, just double checking. So ideally, we would like to maintain a healthy and productive life without spending too many hours practicing and learning tasks over and over again. But we know that this can be too time consuming, and so we can easily lose motivation, mostly because we don't have enough attentional or visual resources for our task. So to continue practicing and improving our abilities. So this is very much a general statement, and it's true for almost everything we do that requires effort. But imagine how difficult it can become when a damage to your brain makes everything, let's say, more complicated, more difficult, and pretty much impossible to perform. So, for instance, a typical example, like in this slide, is visual neglect. So visual neglect is a syndrome characterized by the inability to orient attention to size, control lateral to typically very large lesions to the parietal lobes. So you can imagine how difficult it can be to perform sustained attention tasks. But moreover, no less invalidating is, for instance, another syndrome that you can get from a lesion caused by to a stroke and a lesion to the early visual areas. And in this case, you get cortical blindness, which is pretty much a visual field loss after lesion to the early visual areas. Typically, these lesions can cause either loss of the entire hemifield, or you can have loss of a quadrantic part of the hemifield only. However, no matter how big the lesion is and the deficit in vision, it's still strongly invalidating. So in this talk today, I will try to present some empirical evidence to respond to some of the questions we've been working on in the lab for actually quite a while. So we will try to answer questions of how long can the effect of non-invasive brain stimulation last? And when we do see benefits in what we do those long-term beneficial effects last for a long period of time? And what is the ideal intervention protocol to promote a stable and long-lasting effect, both in the healthy and then eventually translating that into the patient's population? We will also ask whether the manipulations we do that, well, in this talk in particular, we will be concentrating on TRNS, but in the past, we've worked a lot with TMS, and you will also see some TMS data today. The question is whether this effect is more local and selective just for one area, or whether it is widespread across cortical networks. And if we find a network effect, is this effect coherent with the behavioral effect that we measure in subjects? Finally, we will also look at learning effects, and we mostly are interested in these learning effects across multiple sessions, as you have seen nicely before in the morning talks by Daria. And we look at this learning effect as a proof of concept for the potential efficacy of TRNS in promoting a stable and long-lasting behavioral improvements, and also, last but not least, their potential applicability, in clinical efficacy. So in all our work to test all our hypotheses, we use a variety of tools to model a behavior. We use TMS, and more traditionally, we used it in the past. But we also use direct current stimulation. And we typically measure the effect of this stimulation procedure upon cortical networks in the brain using Fmri. So while data on patient, in fact, can tell us a lot about potentially disrupted cortical mechanism, in a more traditional way, we also try to use the classical virtual lesion technique to temporarily mimic those deficits using TMS on healthy subjects. And we do this in order to uncover a mechanism of adaptive or maladaptive plasticity, which is, you just saw talking about this maladaptive plasticity by the previous talk by Ila. And but thanks to this wealth of information, we can try to formulate hypotheses on how to actually translate this into potential recovery for stroke patients. We have more recently started to use transcranial random noise stimulation, and because that it has shown its ability to increase cortical excitability and promote behavior in healthy subjects, with the potential translational application in this case as well. So starting from the first question, so in most of the experiments you will see today, we will use a lot of visual attention task. And we're also very much interested in what Ila was talking about before, which is the interhemispheric interaction, inhibition, or excitation. But we concentrate particularly on attention because that's what we do, and on vision. So a typical task that we use in the lab is called multiple object tracking. And in this study, our aim was to test the immediate effect of inhibitory one frequency RTMS upon the cortical IPS during a multiple object tracking. So the multiple object tracking that we normally use in the lab is a sustained attention task. So we ask a subject to track for a few seconds multiple moving little discs, and that they are highlighted at the beginning, and those are the targets the subjects are required to track. And then they start to move randomly. And then at the end of the trial, one item is highlighted, and the subject has to say whether that was a target or not. Let me emphasize that in this case, subjects are tracking bilaterally. So we really are asking both hemispheres to work in this case. And so we can see exactly how the effects are in the two visual fields, and so we can really see the effect of this interhemispheric interaction. So in this study, we actually replicated the effects we found in the past with parietal lesion patients. However, we also saw differences in the effect between the left and the right hemisphere, as you can see in the inset in the individual data circled up on the slide. We actually saw a more robust effect in the left hemisphere and a more variable response in the right. But the basic finding was a detrimental effect on the side contralateral to the side of the stimulation. However, these differences provided some potential hints on how an imbalance between the homologous IPSs could be causing excessive activity in the healthy hemisphere to compensate for decreased ability in the right parietal lobe patients, for instance. And potentially this might be a model of showing how recovery was being prevented in patients. And thank you, Ila, for introducing all the concept of excessive activity on one side needed to be inhibited, but in your case, you did a lot of excitation too lately. Anyway, this was one of the hypotheses we tested in the lab when we designed an inhibitory RTMS protocol, again, inhibitory 1 Hz RTMS, to help relieve the attentional deficit in chronic patients. So we demonstrated that low frequency RTMS over the LTIPS resulted in a strong beneficial behavioral effect. In fact, you can see here how well patients improved. However, the interesting part of this data is that the effect peaked not immediately at the end of stimulation, but 30 minutes later. So there was this benefit, the time course of the benefit that developed across time. It was not immediate, it was just only later after 30 minutes. And this is sort of also kind of motivated other further experiments in a healthy subject, because the idea here was to understand changes in cortical dynamics after inhibitory TMS, because then it could be a potential model of cortical response to a stroke. So Ila did this nice work when showing that when unilateral IPS is temporarily inhibited with TMS, an entire network of areas responds bilaterally. So we showed a boost in activity in what are likely compensatory nodes of the dorsal attention network, but the same areas also show increased functional connectivity when we look at the functional connectivity. So not only a change in dynamics within and across hemispheres, but also change in connectivity across time. But interesting also in this case, this change in connectivity, and particularly an increase in connectivity that we saw in this data, was evident 60 minutes post-stimulation. However, of all this work, there was never a behavioral benefit in healthy subjects. So we know that RTMS can result in beneficial behavioral effect, particularly in patients, but maybe what we need to do in this case is try to see whether we can boost the inhibitory RTMS effect in healthy participants. So this was kind of starting point for us for the next experiment that we did in the lab and that we published in 2018. Because we started from the concept of state-dependent effect. So we know that the effects of TMS can actually strongly depend on physiological and brain state at the time of stimulation. So in this experiment, we decided to exploit this excitation feature of tRNS to use it as a prime to precondition the visual cortex. So this is a relatively simple phosphine study. And in this study, we hypothesized that the addition of the high frequency noise to neurons at rest will bring them above firing threshold, resulting in cortical excitation. And we measured this potential excitation we created prime in the cortex with phosphine threshold. So we had different control conditions. We had the tRNS priming condition and then phosphine measurements. Then we had a nodal TDCS control condition and a sham condition. So it appears that only the tRNS priming condition had a very interesting effect in lowering phosphine threshold, potentially a marker of increased cortical excitability. And this happened for an extended period of time. And interestingly, this had a time course very similar to previous motor studies. This is one of the first studies by Trento and co-workers on precisely priming the motor cortex and then measure motor potential subsequently after the priming of the cortex. So we had this time course that was very similar to this study. So in the next work, we decided, well, can we try to use this feature of tRNS to put the cortex in an ideal state for higher level cognitive functions, such as sustained attention. And so we went back to visual tracking, to multiple object tracking tasks that you've seen at the beginning of this talk. And in this task, Grace in my lab asked a subject to perform the multiple object tracking task at baseline. This task is very interesting because you can really precisely manipulate and find the speed threshold at which each subject can perform at either 75% correct. So you can actually make the task for the same difficulty for every subject. Then you can measure the threshold at baseline. And you can, what we also did here, we use Fmri to individualize the hotspot for each subject upon which we went to perform our cortical stimulation. In this experiment, we applied this multi-methods approach that you've seen introduced in the previous experiments. So what we did, we preconditioned the cortex with tRNS while subjects were doing the task. At baseline, we already had their threshold measurements. So we had subjects perform the task while they were receiving tRNS bilaterally over the parietal cortex. And then we delivered 10 minutes RTMS where they were at rest. And then we asked the subjects to immediately perform the task afterwards. So typically in this task, subjects usually perform pretty well for up to 40-45 minutes, and then performance tends to deteriorate. So our sustained attention abilities tend to go down, and we can no longer do the task at optimal level. What we found, however, when we, and this is what we found when subjects were in the sham condition. However, in the condition where we coupled those two techniques, tRNS and TMS, we saw this nice sustained effect that lasted for up to 90 minutes. So when subjects were preconditioned and then stimulated with TMS, we actually doubled the duration of their sustained attention ability. With a potential applicability in clinics, obviously, and also in the elderly, probably. So we next ask, well, are these effects that we find with tRNS selective, as we mentioned at the beginning? So this is a question Federica tried to answer in my lab. And well, one way to study behavior and cortical changes that develop across time is actually to use a learning paradigm. So we need to move from one session experiment to multiple sessions experiment. In this particular set of experiments, we carefully chose 2 attention tasks that we knew from work in the lab and in other labs that had a strong attentional component to maximize the responses of the IPS, the node of the attention network we are particularly interested. Although in this case, we're going to look at more an extended part of the dorsal attention network. And we use the double task training node to more efficiently induce learning. I'm not going to go into details, but if you have questions later, I'm happy to answer them. So subject train simultaneously on these 2 tasks on an orientation discrimination task, whereby they were asked to say on each given trial whether these 2 Gabor patches were oriented the same or not. And on different trials, subjects were instead asked to judge whether the 2 Gabors were presented at the same time or not. So remember that visually the task is always the same. Subjects are all thresholded in both tasks, so they are performing at their level of ability, but the task is extremely difficult to do. And so what is changing is the actual task. So then Federica ran 2 Fmri sessions before and after 4 days of short training session, during which we delivered high frequency tRNS, and we had 3 groups of subjects, either the parietal group subjects where tRNS were delivered over the bilateral parietal areas. Then we had middle temporal area active control group where tRNS was delivered over MT, and then we had a sham control group. And this is just an example of one of the trials. I hope you can see it, but the question is, were those 2 Gabor same or different orientation, or were they presented simultaneously or not? So, the first interesting result here was that in the orientation discrimination task, we saw this very large increase in performance after only 4 days of training in this very difficult task. Remember that tRNS was always coupled with behavior, and Fmri was only performed at the beginning and at the end. So this is the behavior result only. And this big improvement was not present in the other 2 conditions, the sham nor the MT condition. This was for the orientation discrimination task. However, in the other task, what happens is that we didn't see any improvement at all. So remember that we did not tell the subjects to train on one task only and ignore the other. They were told to train in both, but for some reasons, the improvement was only for the orientation discrimination task and not for the other task. So bear in mind that the lack of learning is not surprising here because the training was very short. However, improvement in the parietal group was definitely very strong, and we make sure this was not driven by outliers or anything strange. So it was really effective and really strong. And if you want to look a bit into detail of the orientation discrimination task, as you can see here, you can see this very nice progression of learning across daily sessions for the parietal group only and nothing for the other two groups. So, well, this is interesting, well enough, good enough, but what happens in the fMRI? So we look at several networks in the brain, and so, but we concentrated in particular on the dorsal and the ventral attention network, and all the way from the more posterior to more anterior nodes. Functional connectivity was computed at rest, and we compared functional connectivity before and after training for all the three groups. So what you will see is are the differences between the pre and the post training. So these are the results. Connectivity between the nodes of the for the MT and the sham condition did not change between baseline and post training. As you can see, everything is pretty much overlapping before and after, as shown by the purple color. However, we found a strong change in functional connectivity within the parietal nodes in the parietal condition. So indicating stronger connections after training and very selective only for that condition. If you want to look at the brain, so these are all the connections that were stronger after training coupled with stimulations. So in this case, lines connecting the nodes indicate a significant change in connectivity between pre and post training, something that was not there for sham or the HMT condition. But interestingly, we correlated changes in behavior as a function of changes in functional connectivity. And particularly for those subjects in the parietal group, better performance positively correlated with higher connectivity. So next, we ask whether this learning that we see for higher level cognitive functions, such as visual spatial attention, would also apply to lower level visual areas and tasks. We knew from our previous work that TRNS has an enhancing effect upon the visual cortex from the Fossin study, and also increased excitability when we use a priming paradigm. So maybe we could try to leverage these features by using them to speed up perceptual learning, because the ultimate goal is to also exploit TRNS and translational potential in patients. Motion coherence discrimination is a classical paradigm widely used in psychophysics, both in humans and non-human primates. It's called motion coherence indeed. And in this task, subjects are asked to report the motion direction of coherently moving group of dots embedded in noise, which I hope you'll be able to see here in this video. Performance is typically measured as the lowest number of dots subjects can report with an accuracy of about 75% correct. And we know that subjects can improve in this task across multiple sessions, but typically long training are required for an improvement to become significant. So, however, we chose this paradigm because it is widely used, tested, and its effect is, has been replicated multiple times. The task also can be psychophysically manipulated, so we can individually measure threshold to motion discrimination performance, and so it can be used in perceptual learning. So in this set of experiments, we hypothesized that its enhancing properties we had already seen before in our previous work we decided that TRNS could speed up and maybe boost performance. So subjects in this experiment were divided in several stimulation conditions. Actually, precisely, there were 5, but they all underwent 10 days of training with the motion coherence task, coupled with TRNS, high-frequency TRNS. Importantly, we called them back 6 months later and we checked their performance relative to the last training session. So training was performed with daily sessions with concurrent TRNS. So this was about 20 minutes a day of training. TRNS stimulation was done at 1 milliampere for each subject, and we had these 10 days of training. So what did we see at the end? We had several control conditions, as I mentioned before, and some of which were combined because they did not differ significantly. The interesting effect that we saw was that the amount of learning in the TRNS group was substantially bigger, twice as big compared to the other 2 conditions. And more importantly, I would say, performance was retained at 6 months from the end of the training. It was pretty much the same strength as it was at the beginning, right at the end of the training. So we found this effect particularly interesting and compelling. And also we decided to see whether we could actually use this effect and translate it into the clinics, because this is exactly, this work was done in collaboration with our collaborator at the University of Rochester, Chris Laxlin and Tadin, and they have used this paradigm with patients quite extensively, with cortical blind patients. So the question is, can we test it in patients and see whether we can see any efficacy with this TRNS coupled with behavioral training. So cortical eagle blind patients that we mentioned at the beginning, we know that they can be trained on motion coherence within their blind field. And we know that the benefit of training in one task can actually transfer to non-trained task, which is an important feature. However, the problem with this task is that it takes a very long time to get significant benefits with patients. So this makes basically the procedure quite inaccessible for the majority of these patients. So we decided to test these cortically blind patients who did the exact same training as the healthy subjects. And we tested in particular chronic stroke patients affected by unilateral lesion to early visual areas, affecting the opposite hemifield. There was variability in lesions and hemifield deficit, but we measured them with visual perimetry, of course. These patients are also often will show a blind sight. That is, they can correctly report stimuli that are presented in the blind field, despite not being consciously aware of these stimuli. So what we did with them, we trained them on the border of their blind field. So basically, we first presented the stimuli, the motion coherence discrimination task in the good field, and then we slowly moved the task within the blind field. As soon as we hit the spot on the border where we knew they were not performing properly, we started to train that location, and that's the only location we trained. And of course, we use visual perimetry to localize the exact location of the blind area. And we trained them exactly the same as we did for 10 days with healthy subjects. So we tested 11 patients and 3 received active TRNS. 2 received sham stimulation, and 6 patients were trained with the behavior only in the motion coherence task. And here are the results for you to look at. So in the sham condition and in the behavioral condition only, we did not find any improvement across these 10 sessions. And we were not even expecting it to happen, because based on our experience and from the group in Rochester, we knew that 10 days were not enough to learn anything or to improve. However, those patients that were in the tRNS active stimulation condition improved significantly in only 10 days. So this sort of providing an important proof of concept of the procedure on patients, because we sort of dramatically reduce the recovery time in these patients. There is still a lot more to do. These are just 3 patients. So we just recently received support to continue work on this neurorehabilitation procedure for cortical blindness. And we also matched up with some collaborators at Harvard Medical School, where they will be doing some physiology in animals, in mice in particular, creating a stroke model and doing the same procedure in the mouse. So it will be interesting to see what are the cortical and or subcortical structures that might be helping the recovery in the patients, and it will be in the mice, obviously. However, to wrap up my presentation today, as a summary, we question how long can we see this benefit last, and we have seen then combining TMS with tRNS in even one session only, we can prevent the deterioration of sustained attention. In the subsequent studies, we have seen that tRNS can allow this sustained effect and improvement to be measured even at 6 months past the end of training. The next question was, do we see any selective effect? And in fact, indeed, we see this very strong change in functional connectivity within the dorsal and ventral attention network. And interestingly, this change in cortical in functional connectivity is also correlated with behavior. And we also see a behavioral selective effect where we increased the ability in one behavior and not in the other. Did we see any learning? We definitely see a lot of learning during this multi-session tRNS coupled with behavior. So we can definitely tell that tRNS is boosting the learning in all these conditions we tested, both in sustained attention as well as in lower level visual tasks like motion discrimination. And last but not least, again, we started to notice that tRNS actually can speed up recovery in cortical blindness. And I am assuming that this could also be a protocol that can be applied potentially to other clinical conditions, including stroke patients affected by visual neglect, as we mentioned at the beginning. With this, I would like to thank all the people in my lab. This work would not be possible without them, of course. My collaborator at the University of Rochester, collaborator at University of California, Irvine, and all the founding agencies that have supported our work. Thank you.